So we move on to the exclusive mitochondrion club. Pyruvate was the final product of glycolysis. It's what's left of glucose after glycolysis is two molecules of pyruvate. And that all happened in the cytoplasm, but now that pyruvate needs to get into the mitochondrion, which is kind of like an exclusive club. Pyruvate can't get in on its own, so it has to go through this process called pyruvate oxidation. And here's what happens. First, pyruvate can't get in the club. It needs to be modified to get in. It needs to lose some weight. It loses some weight in the form of a molecule of carbon dioxide. So it went from being a 3-carbon molecule to being a 2-carbon molecule, and lost one of those molecules as carbon dioxide. And that's part of where the carbon dioxide in our breath comes from, is from this pyruvate oxidation. Then it needs to pay. It needs to pay electrons to NAD+, put them on the electron bus to go to electron camp. So we've lost a molecule of carbon dioxide. We have put some electrons on the electron bus. Then, finally, in order to get the exclusive mitochondrion club, it needs to join up to a coenzyme called coenzyme A, which I mentioned when we talked about coenzymes a few slides ago. Coenzyme A is a molecule that will introduce what is left of pyruvate to the Krebs cycle, which is like the dance that goes on inside the exclusive mitochondrion club. And as acetyl coenzyme A, or just acetyl-CoA, it enters into the Krebs cycle. So here is an image showing what's really happening in all of this entry into the exclusive mitochondrion club. Here's your 3-carbon pyruvate molecule. It's going to lose that carbon dioxide. It's going to lose some electrons. It's going to associate with coenzyme A, which is a fairly large molecule, shown here just as a word, the phrase coenzyme A. So it associates with this, what's left, this 2-carbon molecule that's left, through a sulfur bond. That's why that S is there. So acetyl-CoA is now ready to go on to the Krebs cycle. So the Krebs cycle has a number of different names. It's also called the citric acid cycle. Sometimes it's called the tricarboxylic acid cycle, because there's a lot of carboxylic acids in there, at least 3, I guess. But this happens in the inside the innermost part of the mitochondrion, the matrix of the mitochondrion. And it's got these 8 different steps that you don't need to memorize, just to sort of familiarize yourself with what's going on here. It's kind of like glycolysis, in that you've got all these different molecules that get transformed one into the other. It's a metabolic pathway that goes in a circle. And so let's see, we've got citrate, isocitrate, blah, 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 blah. So I remember this by remembering the mnemonic, can I keep some sugar for my oatmeal? And there are other mnemonic devices that you could use, but we don't need to. What we really need to remember about the citric acid cycle is this. So here's pyruvate oxidation again. Remember, pyruvate loses the carbon dioxide, pays in some electrons, forms acetyl-CoA, and that's what gets introduced to citrate. And all those 8 steps happen in here, but what comes out of the citric acid cycle, or the Krebs cycle, is the other 2 molecules of carbon dioxide from the pyruvate molecule. We get 3 more molecules of NADH, electrons on the electron bus, going to electron camp. We form 1 molecule of ATP. And we also get a molecule of FADH2, which is a different electron bus. The electrons that get onto FAD don't have as much potential energy, but they're still useful for going to electron camp. So, important stuff from the Krebs cycle. You get 1 molecule of ATP per turn of the cycle. You get 2, or did I say 3? I guess it should be 3, sorry. 3 molecules of NADH, don't look at that, per turn of the cycle, 1 molecule of FADH2 per turn of the cycle, and we're going to release 2 molecules of carbon dioxide. So if I ask you, where does the carbon dioxide in your breath come from, you should say that it comes from pyruvate oxidation, that preparatory step to get to the Krebs cycle, and then the Krebs cycle. So all of the carbon from the glucose that we consume comes from those 2 steps. But we haven't used any oxygen yet. Oxygen doesn't come in until the very end. So another final note here. For each turn of the cycle is from 1 pyruvate molecule. But in glucose, remember when we split glucose, we had 2 pyruvate molecules. So for each glucose molecule, we multiply times 2 turns of the cycle. So when we say 1 molecule per turn of the cycle, it's 2 molecules per glucose that we started off with. 6, not 3 molecules of NADH, 2 molecules of FADH2, and 4 molecules of carbon dioxide per turn of the cycle. Okay, so the last stop, we've gotten rid of all of the carbon from glucose. We've extracted all of the useful electrons. We've made, we've netted 4 ATP from molecules from that glucose. But we've got a last step, and that is the electron transport chain and oxidative phosphorylation. Electron transport chain is probably easier to remember. Now, NADH and FADH2 are going to take those electrons that we've been taking away from glucose and what's left of glucose in glycolysis, pyruvate oxidation, the Krebs cycle, and they're going to drop them off on the electron transport chain. And then, freed of those electrons, everybody's off the bus, so now they're back to going to be NAD plus and FAD, looking for more electrons. And electron camp sounds like a fun place to go, doesn't it? Well, it turns out that it's a work camp. Those electrons that we harvested from glucose have a lot of potential energy. Those are very energetic electrons. Electron camp is actually a series of multi-protein complexes that are embedded in the cristae, all these folds inside the mitochondrion, in the inner membrane. All folded up for extra surface area. So these multi-protein complexes pass the electrons along, and those electrons are going to convert that potential energy that was stored in them into chemical energy. And it looks kind of like this. So what are we looking at in this diagram besides a bunch of purple blobs? These are the different multi-protein complexes. And you can see there's a lot of alphabet soup in here, FMN, coenzyme Q, et cetera, all these different cytochromes. But what we should note is that here's NADH dropping off its electrons and then being released as NAD+, the electron bus, going out to find more electron campers. FADH2, that came from the Krebs cycle, its electrons can't get on the into electron camp on multi-protein complex 1. They have to enter at multi-protein complex 2 because they don't quite have enough potential energy. So here's potential energy is on this axis here, and you can see it's dropping. And as those electrons move through these multi-protein complexes, those electrons are going to end off at the end of the 4th multi-protein complex down here. So they're doing work. What is the work that they are doing? The electrons are being put to work to pump protons into this intermembrane space. So inside the matrix, the pH is relatively high, relatively not basic, but lower in acidity, fewer protons. So all the protons are getting pumped into this space here. So there's a very low pH, very acidic in this intermembrane space. And that produces a pH gradient. pH gradient. So that's why we had this whole chapter talking about membranes and how great they are, and diffusion and osmosis. It's part of the reason. It's basically, instead of protons, think like water. It's being pumped up to the top of a dam. And then those protons can be used to do work. We're generating this gradient, so it's kind of like we put a dam in a river, and we can install a turbine and generate electricity or a form of energy. What's happening in the mitochondria is something very similar. These protons want to correct the gradient. They want to flow downstream. And the only way they can get back into the matrix is by passing through an enzyme that is called ATP synthase. ATP synthase is a really cool enzyme. This is ATP synthase right here. I could just stare at this for a long time because it's so cool. It actually is like a little turbine. It's spinning around. So what's going on in this picture is protons actually enter. This is the inner membrane space. This is the matrix up here, and here's the membrane of the crista. So protons are trying to get back into the matrix. And as they go through, they're passing this like turnstile that spins, and then you have this molecule up, this part of the molecule up here, the protein, that has ADP, adenosine diphosphate, and free phosphate groups are going into these knobs, and they're just getting rammed together to produce ATP. So this thing produces boku ATP. This is where the majority of the ATP that you're using right now comes from. Is from this cool little turbine molecule. It's just ramming ADP and phosphate groups together to produce ATP. It all happens because there's this proton gradient. Protons in here are like, hey, it's really crowded. We want to go and sort of get more space. So they will pass through this turnstile and turn this ATP synthase molecule. So at the end of the electron transport chain, those electrons are depleted in their energy. They don't have very much potential energy anymore. So who is going to accept these tired, low energy, poor electrons? Well, who have we talked about as being very accepting of electrons? Well, maybe I've used the term greedy for electrons, and that would be oxygen. Oxygen is very electronegative, which means it will accept electrons pretty much any condition, even if they're really kind of played out. So oxygen accepts those electrons, and two of those protons that got passed back into the matrix to form, well, what do you think happens if we add protons and electrons to an oxygen atom? We get water. And that's what you see down here, is that those electrons have gotten passed from from the electron bus to the multi-protein complexes, and eventually they're going to get back into the matrix of the mitochondrion. They're going to join up with a water atom from half of an oxygen molecule, oxygen gas molecule, and form water. So the oxygen that we breathe in gets converted to water. So you may have always thought, oh, I breathe in oxygen, I breathe out carbon dioxide, and they're getting exchanged evenly. Well, it turns out that at the cellular level, there's very different processes that we use to consume the oxygen and to produce the carbon dioxide.